Hello everyone and welcome to the biology lecture series, and today we'll be studying the topic sexual reproduction in the flowering plants. Okay, so in the previous lecture what we have studied about the pre-fertilization and the post-fertilization events. Okay, so let's summarize it again. First are the pre-fertilization events. So pre-fertilization events is followed by the fertilization, and fertilization is then followed by post-fertilization events. Okay, this is clear. So during pre-fertilization, what happens? There is the formation of the gametes, okay, which is known as gametogenesis. So pre-fertilization involves the formation of the gametes. So there is one male gamete, another one is the female gamete. Okay. So male gametes, these are formed by the pollen grains. Male gamete, these are formed by the pollen grains. So if you remember, the pollens are usually released in the two-cell stage. Okay, so there is one big cell which is known as the vegetative cell, and another one is known as the generative cell. Okay, so this generative cell further divides, okay, to form two male gametes. Now another one is the female gamete. Okay, you know sexual reproduction, it involves two parents. Okay, so we can say it involves two parents and it involves two gametes. One is the male gamete, another one is the female gamete. So this female gamete is the egg cell. Okay, egg cell you can say, or you can say the ova. So this is produced within the female gametophyte. Female gametophyte is the embryo sac. Pollen grains are the male gametophyte. Alright. So we can write here pollen grains. Pollen grains, this is the male gametophyte. And what is the female gametophyte? Embryo sac. Okay. Embryo sac is the female gametophyte. Now this egg is produced within the female gametophyte. Clear? And then what happens? There is a union of these gametes which is known as fertilization. So the union of gametes is known as fertilization. Clear? So we have studied about the double fertilization which takes place in the flowering plants, which are angiosperms. So one of the male gamete fuses with the egg cell, which forms the zygote, and another male gamete fuses with the secondary nucleus, which is formed by the fusion of the two polar nuclei. And this results in the formation of the endosperm. Okay, so endosperm is a triploid structure. Clear? Now, this fertilization, this involves syngamy, okay, and the triple fusion. So we have studied about the double fertilization. Okay, double fertilization involves syngamy as well as the triple fusion. Now, what happens after the fertilization? What are the structures that are formed after fertilization? Okay, so we'll discuss all of these under the topic post-fertilization. What are the structures that will be developed after post-fertilization? So because of the syngamy, which is what? Fusion of the male gamete With what? With the egg cell. Both are haploid. So what will be formed? Zygote will be formed, which is a diploid structure. Okay. So zygote will further form the embryo. It will form the embryo. Then we have the triple fusion. What is triple fusion? Where the another male gamete it will fuse with what? It will fuse with the two polar nuclei. So these two polar nuclei, these are known as the secondary nucleus. Okay, and this will result in the formation of primary endosperm cell. Okay, PEN is primary endosperm cell, which consists of primary endosperm nucleus. Okay, primary endosperm cell consists of primary endosperm nucleus. The primary endosperm cell will form what? It will form the endosperm. The primary endosperm cell will later on form the endosperm, which is a triploid structure. Clear? So let's start with this post-fertilization event. What are the structures and the events that are formed? Okay. So following the double fertilization, events of the endosperm and embryo development, as well as the maturation of the ovules into seed and ovary into fruit, these are collectively termed as the post-fertilization event. So what is happening during the post-fertilization events? First, there is the endosperm development. Okay, so endosperm develops first. Okay, and then the embryo will develop. So endosperm development precedes the embryo development. Clear? So endosperm development takes place from the primary endosperm cell. Primary endosperm cell, which is diploid, this will give rise to a triploid endosperm. Now, what is happening? Here, next one is the embryo formation. Embryo formation we have seen. Okay, this takes place by the fusion of one of the male gamete with the egg cell. Then what happens? The ovules, the fertilized ovule, they mature into seeds. They get transformed into seeds, and the ovary, after the fertilization, it develops into the fruit. Okay. And let's talk about the endosperm now. So endosperm development precedes the embryo development. Why? Because endosperm is a nutritive structure. Okay, the cells of the endosperm, okay, they contain the reserve food material, which is utilized by the developing embryo. That's why endosperm development precedes the embryo development. Now, the primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. So we have seen the primary endosperm cell, okay, which consists of what? Which consists of the primary endosperm nucleus. Okay, which will be triploid. This divides repeatedly and form a triploid endosperm tissue. Now, cells of the tissue are filled with the reserve food materials and are used for the nutrition of the developing embryo. So, developing embryo utilizes the nutrition from the endosperm. Now, in most common type of endosperm development, what happens? The primary endosperm nucleus undergoes successive nuclear division. Okay, primary endosperm nucleus is triploid. So this will undergo successive nuclear divisions, okay, without undergoing cell wall formation. So this type of nuclear division, where the cell wall formation doesn't take place, this is known as the free nuclear division. Clear? So primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. Clear? So division is taking place, the nucleus is dividing, but there is no cell wall formation. So in one structure only, there are a lot of nucleus. Okay, so within one structure, these nucleus are floating here in a common cytoplasm. So these are all nuclei which are floating in the cytoplasm. Clear? Now, later on what happens? Cell wall will develop. Okay, later on cell walls start developing around this nucleus. Okay. So the stage of the endosperm development is the free nucleus. The first stage is the free nuclear endosperm. So this is the free nuclear endosperm. Okay, let me draw it again. Like this. These were the initially nucleus. So this is the free nuclear stage. I'm just drawing a rough diagram here. Then later on what happens? Subsequently, the cell wall formation occurs and the endosperm become cellular. Clear? So what is happening here? If this is the structure, here were the free nuclei. Now the cell wall formation will occur. Okay. So this is the cellular stage. This is the cellular stage of the endosperm. Now the number of free nuclei formed before cellularization varies greatly. Example, a coconut water, okay, from the tender coconut that you are familiar with is nothing but free nuclear endosperm. So coconut water that you drink, this is nothing but this is a free nuclear endosperm. So it has a lot of nutrition, a lot of minerals, vitamins. Okay, so this liquid part, okay, this is also known as the liquid endosperm, free nuclear endosperm. This is also known as the liquid endosperm. And it provides nutrition. Okay, it is free nuclear endosperm means it consists of thousands of the nuclei, and this is surrounded by a white kernel that you also eat. Okay, and this white kernel is nothing but this is the cellular endosperm. Okay, clear? Now, the endosperm may be either completely consumed by the developing embryo, for example, in pea, groundnut, and beans, before the seed maturation, or it may persist. Okay, this endosperm may persist in the mature seed as well, such as castor and the coconut, and this is used up during the seed germination. Clear? So either the endosperm can be completely consumed before the seed matures, but sometimes it may persist also in the mature seeds. Okay, and this endosperm can be later used up during the seed germination, because seed germination also require, okay, a lot of energy. So this energy is provided by the endosperm, or this nutrition is provided by the endosperm. Now let's talk about the embryo. The embryo develops from the fusion of the male gamete with the egg cell. The embryo should be a diploid structure because both of the gametes, these are haploid and they fuse to form the zygote, and the zygote will form the embryo. So what happens? This is the male gamete. This fuses with the female gamete. Then what happens? Both of them are haploid, and this forms a diploid structure known as the zygote. This zygote is a unicellular diploid structure. You should remember that. It is a unicellular diploid structure. Okay. Then the zygote will further form the embryo. Clear? An embryo will later on form the new individual. Alright. So, Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Okay, what does this mean? So you must have seen the structure of the embryo sac. So this is the, what we are drawing? We are drawing the embryo sac of the angiosperms. So if this is the micropylar end, this is the micropylar end, this is the chalazal end. So at the micropylar end, what is present? The egg apparatus. So this is the egg cell and these are the synergids. Okay. So we can label it. This is the egg cell and these are the synergids. Now these two are the polar nuclei. Okay, and at the chalazal end, what is present? Antipodals are present. Antipodals are present at the chalazal end. So, you know, one of the male gamete, it fuses with the egg cell. So, the egg cell fuses with one of the male gamete. So, zygote will be formed at the micropylar end, or we can say embryo will be formed at the micropylar end. Okay, so here the zygote will be formed, which will later on give rise to embryo. So now you can see how the embryo is formed at the micropylar end. So this is what embryo develops at the micropylar end of the embryo sac, where the zygote is situated. The most zygote divide only after a certain amount of endosperm is formed. Okay, that's why endosperm development precedes the embryo formation, because endosperm is a nutritive tissue. It provides nutrition to the developing zygote and the embryo. Okay, now this is an adaptation to provide assured nutrition to the developing embryo. Okay, now though the seeds differ greatly, the early stages of embryo development, which is known as embryogeny. So embryo development or the stages of the embryo development, this is known as embryogeny. So the early stages of the embryo development are similar in both monocots as well as the dicots. So angiosperm, you know, which are the flowering plants, angiosperm are further divided into monocots and the dicots. So monocots have one cotyledon, whereas dicots, it consists of two cotyledons. Okay. Now let's talk about the stages of embryogeny in the dicotyledonous embryo. Okay. What are the stages of the embryo development? So first is the zygote formation, which takes place after the fertilization. Okay, so this is a post-fertilization event. So fertilization occurs when the sperm cells or the male gamete, it fuses with the egg cells. So both are haploid. And this results in the formation of a diploid zygote. Zygote is the unicellular diploid structure. Clear? Next is the proembryo formation. After zygote, the proembryo is formed. So zygote undergo mitotic divisions to form a multicellular structure known as the proembryo. Now this stage marks the beginning of the embryonic development. Yeah, so you have to keep this by in the development of the proembryo marks, okay, the beginning of the embryonic development. Next is the suspensor formation. So what happens following the zygote formation? The embryo undergoes early developmental stages. On one end of the embryo elongates to form the suspensor. Okay, and this structure anchors the embryo to the surrounding tissues and aids in the nutrient absorption. So this is the function of the suspensor cell. The suspensor cell anchors the embryo. Okay, first role is the anchorage of the embryo to the surrounding tissues. Another one is the nutrient absorption. So it provides nutrition to the developing embryo. Now let's move on. Next stage is the globular stage. Okay, so the embryo progresses into the globular stage then, which is characterized by the spherical mass of cells. That's why it is known as the globular stage. because it is characterized by the spherical mass of cells. Now, at this stage, what happens? The embryo lacks clear differentiation of the tissues and the structure. So, there is no differentiation of the tissues as well as the structure. So, it is just a globular stage. Okay, and the cells are not differentiated. Alright, whenever the cell differentiate, they perform specific function, but at the globular stage, there is no differentiation of the tissue and the structure. So, you cannot distinguish between the different tissues and the structures. Now, after the globular stage is the heart stage. So, heart stage, what happens subsequent to the globular stage, the embryo transition into the heart-shaped stage. Now, here the embryo elongates along the apical basal axis, forming a shape resembling a heart. That's why it is known as the heart stage embryo. Now, distinct region of such an embryonic axis, cotyledon, primordia, and the shoot apical meristem become apparent. So here you can see you can distinguish between the different structures. Okay, so distinct regions such as the embryonic axis, cotyledon, primordia, okay, and the shoot apical meristem, all of these, they become extinct. So you can distinguish between these structures. Now after the heart stage, you have the mature embryo. So what happens through the continued growth as well as the differentiation? Okay, so due to differentiation leads to the specialization of the cells. The embryo matures into fully formed structure with recognizable tissues and the organs. Now the shoot apical meristem give rise to the future shoot system. Okay, including stem, leaves, and the apical buds, while the root apical meristem initiate the root development. Now the cotyledons also, which are known as the seed leaves. What are cotyledons? These are also known as the seed leaves. They also develop fully and the embryo assumes a mature forms ready for germination and seedling establishment. So what you can observe in the mature embryo, you can observe the shoot apical meristem, which will further give rise to the shoot system. Okay, the shoot system includes what? It includes the stem, leaves, apical buds, okay, and another one is the root apical meristem. Root apical meristem, this gives rise to the root system. Okay, which consists of the root and the root hairs and all. Okay, apart from this, you can also see the cotyledons. Cotyledons, these are known as the seed leaves. These are known as the seed leaves. Okay, so all these structures is visible in the mature embryo. Apart from this, you can see the embryonal axis also. You can see the hypocotyle, epicotyle. Okay, all of the structures you can see. Now these are the stages. So this image depicts the stages of the embryogeny in a dicotyledonous embryo. So we are talking about the dicots here. So in this image, you can see, so this is the condition of the embryo sac after the fertilization. Okay, what is A? This is the condition of the embryo sac after the fertilization. So here you can see after the fertilization, zygote is formed at the micropylar end. So this is the micropylar end. Okay, this one is the chalazal end. Okay, at the micropylar end, the synergies are present and the XL fuses with the male gamete to form the zygote, and the synergids, they degenerate. Now, this is the primary endosperm cell, which is formed from the central cell. So, central cell give rise to the primary endosperm cell. Okay, this primary endosperm cell consists of the primary endosperm nucleus, which will be diploid. Here, now these are the degenerating antipodals. So antipodals, 3 antipodals are present at the chalazial end. But what happens after the fertilization? The antipodals also degenerate, the synergies also degenerate, zygote will be formed. Okay, a zygote will be formed. Okay, and antipodals and synergies will degenerate. Clear? After this, you can see here the zygote. Zygote is formed and zygote divides to form this proembryo. Clear? And then you can see proembryo will give rise to the globular embryo. Here you can see the suspensor. The suspensor cell aids in the nutrition. It aids in the nutrition also, and it helps in the anchorage of the embryo to the tissues. Clear? So this is the heart-shaped embryo. Okay, you can see it is in the shape of the heart. That's why it is given the name heart-shaped embryo. In globular embryo or in the globular stage, what happens? You cannot differentiate between the different tissues as well as the different structures. But in the heart stage, you can differentiate. Okay, finally, you can see this is the mature embryo. In the mature embryo, what you can see? This is the radicle. So radicle will give rise to the root. And the plumule, it will give rise to the shoot. And here you can see the cotyledons, which are known as the seed leaves. So in the dicots, if we are talking about the dicots, then 2 cotyledons are present here. So this is how a mature embryo will look like. A mature dicot embryo. These are the 2 cotyledons. Okay. So in the mature embryo, you can differentiate between the different structures. Clear? Now, so this image, let's talk about this image. So this is a typical dicot embryo. And what does it consist of? It consists of the embryonal axis and the 2 cotyledons. So here you can see plumule, this will give rise to the shoot. Radicle will give rise to the root. And this is the root cap. So root cap is the protective covering. Okay, it protects the root tip. Okay, from the degradation, because the roots, they are underground. They grow below the soil. Okay, so soil is very hard, soil particles. So this root tip can get ruptured. So to prevent the rupturing of this root tip, root cap is present. So it protects the root tip from the damage by the soil particles. This is the hypocotyle, which is the region below the cotyledons. Okay, then we have the epicotyle, which is the region above the cotyledons. Okay, now let's discuss about this. A typical embryonal dicotyledonous embryos consist of the embryonal axis. What is embryonal axis? The central axis containing the hypocotyle, which is also known as the embryonic stem. Okay, and a radicle, which is known as the embryonic root. So hypocotyle is known as the embryonic stem, whereas the radicle, this is known as the embryonic root. From this, we have the cotyledon. What is cotyledon? These are the 2 seed leaves that store the nutrients for the initial growth. So cotyledons, what is the role of the cotyledon? They store the nutrients which are required for the initial growth. Then we have the hypocotyle. What is hypocotyle? Hypocotyle is the embryonic stem located just below the cotyledons, which connects the root to the shoot system. What is epicotyle? Epicotyle is a region above the cotyledon. So these are the structures that you can see in the typical dicotyl embryo. You can see the embryonic axis, you can see the cotyledon, you can see hypocotyle, epicotyle, you can see the radicle, as well as the plumule. Okay, you can see plumule also. Plumule will give rise to the shoot. Radicle will give rise to the root. Okay. Now let's talk about the structure of the monocotyledonous embryo. So this is the image showing you the structure of the monocotyledonous embryo. So you can see what are the structures here. Here you can see this is the radicle which will give rise to the root. Okay. Root cap you can see which will protect the root tips. Coleorhiza. What is the coleorhiza? So it is the sheath that protects the root tip or the root apex. Okay, similarly, coleoptile. So, coleoptile is a sheath that protects the shoot apex or the shoot tip. Okay, this is clear. So, this is the shoot apex you can see. Okay, scutellum. Scutellum is nothing but it is a degenerated cotyledon. So, monocots, you know, it consists of only one cotyledon. Clear? So, these are the structures which are present in the monocotyledonous root. Let's talk about the main parts of the monocotyledonous seed now. Okay, we are talking about the seed. It consists of the seed coat first. Seed coat is the protective outer covering of the seed. Okay, then the seed consists of the endosperm. Endosperm is a nutrient-rich tissue surrounding the embryo, providing nourishment during the germination. Then embryo. Okay, so in the previous slide, we were talking about the embryo. Now we are talking about the seeds. Embryo, the developing plant contained within the seed, consisting of the embryonic axis, which is the radical, hypocotyle, and the epicotyle, and one or two cotyledons. So monocots, they consist of one cotyledon. Dicots, they consist of two cotyledons. Clear? Then the embryo, we have already seen. Okay, it consists of this embryonic axis, cotyledons. So these are the leaf-like structures, also known as the seed leaves. So these store and provide nutrients to the developing embryo. Then you have the radical. Radical is the embryonic root that emerges first during the germination. So radical will give rise to the root. Then you have the hypocotyle. Hypocotyle is the part of the embryo between the radical and the cotyledon. So it is present below the cotyledon, which may elongate to lift the cotyledon above the soil during the germination. And what is epicotyle? Part of the embryo above the point of attachment of the cotyledon. So it is the region above the cotyledons which may develop into the root system. Okay, so these are the main parts of the, you can say, monocot seed. Okay, what does embryo consist of? Embryo consists of this. Yeah. So in this lecture, what we have studied about the post-fertilization event. So what are the post-fertilization event? First is the formation of the endosperm, then the embryo is formed, the ovules finally mature into transform into seeds and fruits, and ovary transform into the fruit. That's why in angiosperm we say the seed is enclosed within the fruit, or you can say ovule is enclosed within the ovary. Clear? So we talked about the structure of the embryo for both dicots and monocots. Okay, and then we also talked about the stages of the development of the embryo in case of the dicotyledonous embryo. Okay, so I hope you enjoyed this lecture. We'll meet you in the another lecture. Thank you very much for listening.